Today we will deal with heme catabolism. Okay, heme catabolism. In previous class we studied about heme synthesis. Now we will deal with heme catabolism. This is the heme, right? You know the structure of heme. There is a central ferrous atom. There is 4 pyrrole rings around the ferrous atom. Simply the heme catabolism is heme is converted to unconjugated bilirubin, then it become conjugated bilirubin and it is excreted. This is the simplest form of heme catabolism steps. First, heme converted to or heme catabolized to unconjugated bilirubin. See this is a linear molecule, right? This is a circular molecule. So this is a linear molecule. First it become linear molecules, then it is conjugated and converted to conjugated bilirubin. Why it is conjugated? Because this unconjugated bilirubin is water insoluble, so it become water soluble by conjugation and it is excreted. This is the simplest form. We will deal with again differently. Okay, this is the steps in detail. This is the steps. There are 9 steps for heme catabolism. Okay, first step is generation of bilirubin, how this heme is converted to bilirubin. Then second step is this converted bilirubin is transported to liver, and third step, after transport to liver, it should be uptaken by this liver cells. So the third step is hepatocellular uptake. Then in the hepatocytes there should be a step like intracellular binding. Okay, then conjugation. Then sixth step is excretion of bilirubin to bile. After conjugation liver, it is excreted into bile. Then seventh step, fate of conjugated bilirubin in intestine. This is bilirubin. After excretion to bile, this bile is secreted into intestine. So some changes are occurring in the intestine. Eighth step, enterohepatic circulation. Some of this converted bilirubin, that is fate of bilirubin intestine, it is somewhat it is uptaken into enterohepatic circulation and final excretion. These are the steps. We will deal in detail everything. First step is generation of bilirubin, right? 300 milligram of bilirubin is formed per day. 80% of bilirubin is from hemoglobin of old RBC. Old RBC after 120 days is catabolized. So HB is released. This HB is catabolized to bilirubin. So 80% of the bilirubin is from hemoglobin of old RBC. 10% of bilirubin is from immature RBC or ineffective due to ineffective erythropoiesis. There will be some immature RBC. It is catabolized into bilirubin. Remaining 10% from cytochromes myoglobin. You learned that some heme containing protein other than hemoglobin such as cytochromes, myoglobin, etc. 10% of bilirubin is from cytochromes and myoglobin, etc. Okay. The site of generation of bilirubin is reticuloendothelial system of liver. Okay, reticuloendothelial system of liver, spleen and bone marrow by microsomal heme oxygenase system. This generation of bilirubin is by microsomal heme oxygenase system. So heme oxygenase is present in the reticuloendothelial system of liver, spleen and bone marrow. So if there is a question like where is heme oxygenase is present, it is in RES or reticuloendothelial system. So the formation of bilirubin requires molecular oxygen and NADPH. Okay. Cytochrome C also required. Heme is derived from heme containing proteins such as hemoglobin. Hemoglobin converted to globin and heme, right? Hemoglobin is converted to heme and globin. The first step. What happened to this globin? Globin is a protein, so it is catabolized to amino acid. This amino acid is released into the blood and it is reutilized to form hemoglobin. Okay. What happened to this heme? We will deal with it. By the time heme reaches the oxygenase system in the reticuloendothelial system, iron is in ferric state. Okay. Normally, iron is in ferrous state. So, when the heme reaches the oxygenase system, this iron is in ferric form or hemine. Okay. This heme is known as hemine. Hemine reduced to heme. This is the hemine, the ferric, where iron is in the ferric form. This hemine is converted to heme again by conversion of NADPH to NADP. Okay. The second step, using more NADPH and oxygen, it is added to methynyl bridges between pyrrole 1 and 2. You know the pyrrole, there are 4 pyrrole links, 1, 2, 3 and 4. Then NADPH and O2 helps the in NADPH and O2 present. So this O2 is added to methynyl bridges between pyrrole 1 and 2. This is the step. See, here is the first pyrrole ring, second pyrrole ring, in between OH is added. Okay. In third step, further addition of O2 break bond between 1 and 2 pyrrole rings and forms biliverdin. This biliverdin is nothing but a linear form of this heme molecule. That is, pyrrole rings are in circular form in here. Now it is converted to a linear form. Okay. In this step, iron and carbon monoxide are released. Okay. This is the only step where endogenous carbon monoxide is released in our body. This is the hemine form. Iron is released and reutilized, carbon monoxide is released, and this heme is converted to biliverdin 9 alpha. This is a linear molecule. This biliverdin 9 alpha is a green pigment. This is the green pigment. Biliverdin is a green pigment. So in the first step, in effect, the heme is converted to hemine. This hemine is converted to biliverdin, and it is a green pigment. Okay. In birds and amphibians, this green biliverdin is released. Okay, no further steps are there. But in mammals, biliverdin reductase convert biliverdin to bilirubin. Okay, by a reduction. Then biliverdin 9 alpha with the biliverdin reductase converted to bilirubin 9 alpha, which is a yellow pigment. You know, bilirubin is a yellow pigment, biliverdin is a green pigment. So this is the step. Hemine converted to heme for heme. This heme is converted to biliverdin, and biliverdin is converted to bilirubin by microsomal heme oxygenase system present in reticuloendothelial system. Okay. After formation of this bilirubin, first step is the formation of bilirubin. You have seen how bilirubin is formed. This bilirubin is transported to liver. Okay. Actually, this bilirubin formed in RES or reticuloendothelial system is water insoluble. So, this bilirubin is transported in plasma bound to albumin. Okay. One albumin binds with the 2 bilirubin. Okay. One albumin binds with 2 bilirubin, and it is a loose combination actually. If aspirin, penicillin, that's drugs like aspirin, penicillin is an antibacterial, you know. If we eat this aspirin, penicillin, etc., it can displace bilirubin because this is a loose combination with the albumin. And this albumin bilirubin complex reaches the liver. Okay. Now, after reaching this liver, it should be taken into hepatocytes. Okay. Third step is hepatocellular uptake. After formation of bilirubin and transport to liver, it is taken into hepatocytes. At sinusoidal surface of the liver cell, free bilirubin is taken up. This albumin and bilirubin bond is detached, and this bilirubin is taken up into hepatocytes. This is a carrier mediated active process, that is simply facilitated diffusion. Okay. Then fourth step is intracellular binding. Within the hepatocyte, now albumin is detached, right? Then within the hepatocyte, they bind with the glutathione S transferases like ligandins, protein Y, etc. Okay. So glutathione S transferases bind to this bilirubin in the hepatocytes, which is known as intracellular binding. This makes solubilize bilirubin prior to conjugation. Next step is the conjugation. Prior to this conjugation, the bilirubin should be solubilized. For that purpose, in hepatocytes, this bilirubin binds to glutathione S transferases. This prevents efflux back to blood. Now we will move to conjugation. How bilirubin is conjugated. UDP glucuronic acid helps to conjugation. So bilirubin is converted to bilirubin monogluceride in the presence of UDP glucuronic acid. The enzyme used is UDP glucuronyl transferase. Okay. Then again one more UDP glucuronic acid. Glucuronic acid is added by UDP glucuronyl transferase to form bilirubin diglucuronide. This occurs in endoplasmic reticulum. Okay, conjugation occurs in endoplasmic reticulum. Actually, heme oxygenases, heme oxygenase system is present in reticuloendothelial system. In macrophages as the organelle. Macrophages are the organelle in which heme oxygenase is present. In conjugation occurs in endoplasmic reticulum. Drugs interferes with the conjugation. What are the drugs which interferes with the conjugation? That is when these drugs are taken, the bilirubin is not conjugated. What are those drugs? Primaquine, novobiocin, chloramphenicol, androgens, etc. Now the first step is excretion into the bile. Okay, this is an active process against a concentration gradient. So energy is needed. This is a rate limiting step in the bilirubin catabolism. There are 9 steps, Then this excretion into the bile is the rate limiting step, which is a rate limiting step in the heme catabolism. This is the excretion into the bile. This is mediated by ATP binding cassette protein called multi-specific organic anion transporter or MOAT in plasma membrane of bile canalicular cells. So MOAT protein helps in excretion of this bilirubin, conjugated bilirubin into the bile, which is a rate limiting step. Phenobarbital, this is a drug which induces conjugation and excretion. Okay, phenobarbital is the drug which induces conjugation and excretion. So it is given in jaundice patients. Okay, this is the picture showing the heme catabolism and conjugation. You can see. Then 7th step, fate of conjugated bilirubin. What happened to this conjugated bilirubin after excretion to the bile? This bile containing bilirubin, conjugated bilirubin is excreted into the intestine. In intestine, the remaining steps occur. In the intestine or terminal ileum, this conjugated bilirubin becomes free bilirubin. Okay, by beta glucuronidase, which is a bacterial enzyme. There are some bacteria in the intestine. They produce beta glucuronidase enzyme, and this conjugated bilirubin becomes free bilirubin after removal of this glucuronide group. Then this free bilirubin is converted to urobilinogen. Okay. 20% of the urobilinogen is taken up into enterohepatic circulation and it is moved to kidney and excreted through urine, and which is in the amount less than 4 milligram per day. The urobilinogen excreted in urine is less than 4 milligram per day. What happened to the remaining 80% urobilinogen? It is converted to mesobilinogen and stercobilinogen and it is excreted to fecus. Okay. This is the final excretion. Urobilinogen is stercobilinogen is colorless and it is oxidized by atmospheric O2 oxygen to colored compounds urobilin and stercobilin. Urobilin is orange brown and stercobilin is dark brown. Normal color of fecus is due to these compounds. Normal color of fecus is orange brown and it is due to this urobilin and stercobilin. This is the total steps I can explain. First, extravascular or intravascular hemolysis occur and unconjugated bilirubin is released into the blood. This unconjugated bilirubin is water insoluble, so it binds to albumin and it is transported to liver. In the liver, this unconjugated bilirubin is taken up into hepatocytes by before that it should be solubilized with the ligandins or Y protein or Z protein, and after that conjugated with the glucuronidase transferase, and this glucuronidase transferase is glucuronal group is removed by intestinal bacteria releasing beta glucuronidase enzymes, and urobilinogen is formed. In this urobilinogen, this 20% of the urobilinogen is taken up into enterohepatic circulation and moved to kidney, and it is excreted through urine. Remaining 80% of the urobilinogen is converted to stercobilinogen and it is excreted through fecus. Okay. Normal serum bilirubin is 0.2 to 0.8 milligram per deciliter, and direct or conjugated bilirubin is 0.1 to 0.4 milligram per deciliter. Indirect or unconjugated bilirubin is 0.2 to 0.6 milligram per deciliter. These are the normal values. You should study it very thoroughly. And measure this serum bilirubin is measured by van der Berg method. Okay, this measured by van der Berg method. I will explain in the next class what is this van der Berg method and what are the types of jaundice, etc. Now we will move to summary. Thus, heme catabolism occurs in reticuloendothelial system, specifically in the macrophages. Okay, hemoglobin is converted to heme, and globin is catabolized to amino acid, which is reutilized at globin pool for globin formation, and heme is converted to biliverdin in the presence of heme oxygenase. And iron is released, or ferric form is reutilized for heme production. Carbon monoxide is also released here. This carbon monoxide is the only pathway where endogenous carbon monoxide is released. What happened to this biliverdin? This biliverdin is converted to bilirubin. Okay, biliverdin is converted to bilirubin by biliverdin reductase using NADPH. Okay, what this biliverdin reductase do? This biliverdin convert methinyl to methylene bridges, methinyl to methylene bridges, adding oxygen. Okay, bilirubin is toxic, insoluble in water. Bilirubin bind to albumin in the blood. You know, albumin has two types of binding site, high affinity site and low affinity site. 75% of the bilirubin bind to high affinity site, 25% bind to low affinity site, and released to tissues in this 25% release into tissues in pathological condition. Okay, then uptake to hepatocytes by facilitated diffusion, then conjugation by UDP glucuronidase transferase. Then part of conjugated bilirubin is again taken up by hepatocyte by the help of protein OAT1, B1, OAT1, B3. This is organic anion transporter 1 and organic anionic transporter 1B3 proteins. Okay, this any problem with this organic anion transporter protein will leads to interruption in reuptake of this conjugated bilirubin, right? This will lead to Rotor syndrome, in which direct hyperbilirubinemia is seen. I will explain it in the next class when we discuss about jaundice. Okay, then the secretion into the bile, which is the rate limiting step. A secretion into the bile is a rate limiting step. This is with the help of active transporter MRP2, multi-drug resistant protein 2. Okay, as it enters the bile go to ileum and colon. Then by the action of enzyme beta glucuronidase, conjugated bilirubin is converted to urobilinogen. 90% of urobilinogen is converted to stercobilinogen, excreted into fecus. Some part of urobilinogen enters enterohepatic circulation and reaches kidney and excreted into urine as the urobilin. Okay, this is the summary. Thank you so much.